There are 28 participants.
All right.
And we are already in YouTube.
Okay, great.
Ya estamos en vivo, doctora.
Sí, muchas gracias. I'm gonna wait 1 more minute, so
Okay,
people is coming, so.
great.
Well, hello everyone. Good afternoon. I'm very happy and it's an honor for me to introduce you Dr. Donna L. Corral. She's an expert in the field of behavioral neuroscience. She did a PhD in neuroscience in the University of Virginia in Tucson. In the University of Virginia, and she did a postdoctoral position in the University of Arizona in Tucson, and this was in 1994. She was working with Carol Barnes. You know that some of you know. She's a leader researcher in neural mechanisms of learning and memory and brain aging. So because she was doing that position, she starts working in learning and memory and aging. And her subsequent independent work was focused and is focused on changes in neural processes, mediating learning and memory mechanisms. And she has been looking these processes related to various hormonal situations and to diary and aging-related states, particularly across the transition to senescence and health and disease. And most recently, she incorporated the idea of benefits from enriched lifestyles for brain and overall physiological health by directly manipulating exposures to cognitive and physical activity. Her findings suggest that physical and mental activity improve learning through a similar mechanisms, which is BDNF. So that's why I asked her to talk about these new findings that she got in her lab. And what I can tell you is that her contributions in science are main 4 things. And one is the opposite effects of estrogens and learning and memory across adulthood. And you can see that in her first slide. And the second contribution is to, she has been working in multiple memory systems, metabolisms, and aging. And she also has been working by directional changes in and saturation of synaptic plasticity. And one very important thing is that all these studies that she has been doing in animals, she's doing that translation to humans. So it's a very interesting point. She's a member of different societies, and she's also a member of the editorial board of different journals, such as Neurobiology of Learning and Memory, Frontiers in Aging Neuroscience, Hormones and Behavior. And she has been working also in programs for the community. I know that recently, since 2013, she has been working in a program that she developed in movement for healthy aging, and it's a program of dance exercise. Well, she's now a member of the biology department, and she's in the graduate committee. So she works with a lot of students also of the University of Syracuse in the biology department. She has more than 60 articles, I will say. And she has a grant from NIH that is currently now until next year. So this is very good because, you know, with this situation, not all the researchers are support. So she has this program from this grant from NIH, and I'm very happy to have Donna Corral here because she has been very helpful to our students and to our memory and learning field. Recently, she published an article in the Neurobiology of Learning and Memory journal, where we contribute in honor to James McGaugh. And she was working with him at some point of her career. So she has been in contact with a lot of leaders in the field of learning and memory, such as Carol Barnes and James McGaugh. And well, it's an honor for me to have her here because it's very difficult to say yes to a virtual talk. So I'm very happy to have you, and thank you so much, Donna.
Well, thank you, Gina. And actually, Gina failed to mention that she's a special editor for that journal, the edition, the special issue on honoring Dr. James McGaugh. And I thank her for all the hard work it is to edit, to be the head editor for a special issue. Well, and thank you for that really kind introduction. And I would like to start by saying I wish I could give this talk in Spanish, but it would be a very short talk, I'm afraid. So I'm going to have to deliver it in English. And so hopefully I will speak slowly because I typically don't speak very slowly. So hopefully I will. And I'm delighted to be here. I wish I could be visiting you on site, but maybe the next time I speak, I can be on site with you in Mexico. Okay, so as Gina mentioned in her very lovely introduction, that the work that we do and that I'm really passionate about is understanding experience-dependent changes in the brain, experience-dependent neuroplasticity. And in particular, I'm interested in what are the neural mechanisms of learning and memory, especially as they change across the lifespan and with hormonal states. And so all of the work that you see here, the different projects that the lab is involved in, has the theoretical scaffolding that there are extrinsic and intrinsic experiences, and I say that very broadly, that put the brain in a state that is better or worse prepared to process certain types of information. And I think that the shift in processing may reflect the relative participation of different neural systems that we call memory systems, as many of you know and study. And so we actually look at the neural mechanisms through what we call a multiple memory systems lens. And we try to understand the contribution of bioenergetics, such as neural and glial metabolism, cell signaling, and especially trophic factors, which is what I'm going to focus on today. So as you can tell from the title, and as Gina alluded to, I'm going to talk about some of our more recent work that we've taken to test the shared mechanisms of physical and mental activity benefits on subsequent cognition that we're going to call priming. So priming with exercise and cognition, and the role that BDNF signaling may play in those benefits. Okay, so we call it Use It and Boost It. That's the name of our project. And so hopefully by the end of the talk, you'll see why we call it Use It and Boost It. Now, we all know, and there's lots of evidence that experiences actually affect the brain. In fact, a lot of you out there are studying how experiences affect the brain. Now, enriching experiences, some of those experiences may actually impair brain function and some may enhance brain function. And we tend to think of enriching experiences as those that actually enrich function. Now, enrichment or enriching experiences often have a physical component and a mental component. And so we've, over the years, have been trying to tease out, we as a field have tried to tease out the impact of physical activity alone and maybe mental activity alone, but more so the impact of physical activity alone and how they enhance the function of different neural systems or memory systems. And we've come far to understand that physical activity does enhance function and maybe even provide protection against risk for neural disorders or protection against cognitive aging, for example. However, we don't really know that much about the mechanisms and whether physical activity and mental activity have shared mechanisms. And so I'm going to tell you about the work we use using a voluntary exercise paradigm for our physical activity, priming activity, and a spontaneous alternation working memory task for our priming task for mental activity. Again, I'm going to tell you that BDNF, we think, is a factor that enhances function and maybe may lead to eventual neural health and protection. Now, what's interesting is we know a lot more about physical activity, I think, than mental activity, only because I think when we think of what learning actually is priming from previous experiences. So we actually study something and that's a priming event. And then that supposedly helps us with recalling or accessing that information at a later time or processing it, and might help us with decision making. However, that's typically the same type of information that we study is the information that we're learning or recalling. What we're interested in is whether mental activity that can prime the brain for benefits in other tasks, solving different kinds of tasks, okay? And most of this work is done by this former PhD student in the lab, Claire Scavuzzo. She conceptualized this and really started these studies in the lab and actually developed some of the techniques that we now use to continue this work. Now, Claire is now a postdoc at the University of Alberta with Clay Dixon doing very interesting metabolism and memory work. So before we get too far, I'd like to give you the take-home messages. And that is that, so that if nothing else you understand, you walk away with some information. So that I'm going to hopefully tell you that priming with prior physical or mental activity can boost cognition on different kinds of memory tasks. And we think it's doing this through BDNF signaling during the learning, not necessarily the trophic effects of BDNF on the brain, like neurogenesis and angiogenesis and so forth. We feel that I'll show you hopefully that priming augments BDNF release in awake behaving animals. And this actually has never really been done before until Claire came along and developed the methods to do this. And then that BDNF signaling may mediate the priming effects that by regulating one kinase that's active in the brain called glycogen synthase kinase 3 beta, which we know actually can have a lot of damaging effects in the brain. And so we think that BDNF inhibits this activity. And so then the main take-home message is that there's a shared neurobiological mechanism between the physical and mental activity, and that mechanism is BDNF. One of them. I'm sure there are many, many others, but the one that we're interested in studying. So great thinkers and philosophers for many thousands of years have actually been warning us to stay active, both mentally and physically, in order to fight off the frailties of old age. So Cicero said, it is exercise alone that supports the spirits and keeps the mind in vigor. So connecting the body and the mind. And Da Vinci said that iron rusts from disuse, water loses its purity from stagnation and freezes by cold. Even so does inaction sap the vigor of the mind. So we've known this for many, many years. But despite these early warnings, we actually still don't get enough exercise. And so this is from the Centers for Disease Control, that 80% of adults still don't meet the recommended guidelines for the amount of exercise we're supposed to get weekly, at least in the United States. Now more than ever, I think Zoom has made us even more sedentary. For example, I didn't even have to walk from office to office to meet you guys today. I actually am just sitting at my desk working on whatever I was working on before and now coming to give a talk. So we know that this has led to an increase in early onset of chronic diseases, including type 2 diabetes, metabolic syndrome, obesity, that we know are also risk factors for neural disorders such as Alzheimer's disease and Parkinson's disease. Now, sadly, this is also true for younger people, so children and young adults, that there's an increase in obesity, and now it's type 2 diabetes in juveniles, which we had never seen before. So we do know there's a lot of evidence, and I'm going to share with you some of what we are contributing to that evidence for the benefits of exercise, for general physiology, and for brain health, even though we don't know, as I mentioned, as much about brain health. And I'm hoping that someday physical activity could be used, and even cognitive activity could be used as a treatment. So we could actually get a prescription for maybe that spin cycle that we've been wanting to have. So it could actually be covered by insurances, because it may be more effective than some of the pharmacological treatments that we use. So it's really important for health worldwide, because physical activity and cognitive activity is inexpensive unless you're Lumosity and you charge for it, and accessible. So it's usually typically inexpensive and accessible, so making it a great treatment option. Okay, so I mentioned that we look through a memory systems lens, and what do I mean by that? Well, animals actually solve different kinds of cognitive tasks. Again, a lot of you, I don't know who's out there, but a lot of you out there use different kinds of cognitive tasks that actually, they use different strategies to solve these cognitive tasks that tap into different neural systems. And we're going to call those neural systems memory systems. And while, of course, there are lots of neural systems that can be considered memory systems, we use two in particular, because the hippocampus and the striatum, because they readily dissociate based on the cognitive attributes that are used to solve the tasks. And we've developed tasks, we and others have developed tasks that readily sort of express the strategy that the animal is using. And because of lots of studies that have been done tell us that as an essential an output or a reading of the function of the striatum in the hippocampus. So one of these tasks, the striatum sensitive task, and these were actually first developed by Edward Tolman. So we didn't really develop these. And I think, Gina, you have or used the Tolman maze, and others I'm sure have used the Tolman maze. We've simplified this considerably. And what we use is we use a 4-arm, might be called radial maze, and we ask the animal to solve it using what we call a response learning strategy, where the animal just has to make a body turn to solve the task, to find food on the maze. And so if they start here, they would, in this case, would make a right turn. In the next trial, if they were started here, they would make a right turn and the food would be here. And so they would learn to turn this way, essentially. In our hippocampus sensitive task, we use the same maze with the same general paradigm, except the learning strategy the animal uses is to actually go to the same place in the room. And so we call that the go there strategy, or the place learning strategy. And so if the animal was started here, it would find the food there. If the animal was started here, it would find the food there, without regard to the turn needed to get there. So in this case, so this is a hippocampus sensitive strategy, and this response learning is a striatum sensitive strategy. So all of the experiments I'm going to describe today use single solution versions of these tasks, where animals are trained to find food on a maze using either place strategies or response strategies. So those are the two tasks we use to test for the priming effects. Now, what's so powerful about these tasks is, as I mentioned, they both use the same maze, the same general paradigm. They require the same behaviors from the animals, and they are essentially only different because of the cognitive strategy used to solve the maze. So differences in these tasks can tell us about the hippocampus and the striatum without other intervening variables such as motivation and so forth, because they require the same motivation and other factors. Now, there are, of course, many other striatum sensitive and hippocampus sensitive tasks, and I have blatantly left off the other memory systems such as amygdala, some of your favorites, I know, especially Gina and Roberto, I think, and many of you out there, and prefrontal cortex and other brain areas. But it just, as I said, we use these two as a paradigm to test the effects of exercise and cognitive activity or mental activity. Okay. So one of the most common ways we've determined that these memory systems are, that there are memory systems associated with different task attributes is to make lesions. So you can make a lesion to the hippocampus and you find that you get impairment in place learning, but no big effect on response learning. And you can make a certain kind of lesion to the striatum and find an impairment on response learning. And the work of Ray Kesner and Norman White and many, many others, Mark Packard with Jim McGaugh and others, Rob McDonald, have really developed this idea that there are independent systems where you can actually just double dissociate or triple dissociate structures by task. Now, what's interesting is there are also conditions, however, where the lesion of one structure causes an impairment in learning associated canonically with that structure, but also causes an enhancement in the non-canonical task. Okay, so we can see impairments, hippocampal lesions can cause impairments in place learning, but enhancements in response learning. And likewise, for striatum, lesions to the striatum cause response learning impairments, but place learning enhancements. So this idea is that there is competition between memory systems whereby the impairment of one leads to the enhancement of the other. Now, the corollary is also true where you can see that boosting function of one system can actually lead to impairments in the other. Now, this is less common, but it has been seen. Again, the work of Paul Gold, who's my collaborator and my spouse, so he's my partner in and out of the lab, has shown some beautiful work looking at acetylcholine to show this. Okay, so there is competition between systems. So the first question we wanted to ask is whether we see competition or collaboration between memory systems. So exercise has been shown to improve hippocampus sensitive cognition in humans and in animal models. Now, if the hippocampus and striatum compete for cognitive resources, then would exercise impair striatum sensitive learning if there's such big enhancement of hippocampus sensitive learning? So that was the first question we were actually interested in. So we tested this, the effects of exercise on hippocampus and striatum sensitive learning in those two tasks I mentioned before. And again, I need to mention that all of this work is done in males, even though a whole other more than half of my lab studies females. So I want you to know that we are starting to apply some of this work to questions of females and sex differences. So we tested male rats on learning tasks after 3 weeks with or without voluntary access to running wheels. So what we did is we had a sedentary group where they had no access to wheels, where they just sat in their home cage. And we had, an exercise group where they had free access to a running wheel through this little bridge 24/7, so all the time. And we show that over the course of the 3 weeks, they actually learned to use the running wheels. And then actually the third week, when we food restrict the animals, there's a big boost in running. And that's been described over many, many years, decades, that animals actually, when food restricted, they actually increase their activity. And we can talk about why after the talk. So they do run, actually. So we have shown that, so they are exercising. And then we train them on either place learning or response learning. So different sets of animals were trained on place or response learning. So we only do one learning task in all of our studies before we do the priming. So I'm going to show you that when animals are primed with exercise, it enhances place learning, which was expected because we know it's a hippocampus sensitive task and others have shown similar kinds of effects. So if you see here, we show trials to criterion so that the animals that had exercised took fewer trials to criterion, so they were faster at learning this task compared to the sedentary animals. Very simple and straightforward finding. And down here, you can see that their learning curves are actually much more steep in the exercise primed animals, where they actually start out the same, but learn much more quickly. And in fact, the sedentary animals never reach our criterion of 90% correct. For response learning, our hypothesis was not supported. We actually found beautiful enhancement of exercise, our competitive hypothesis, where we see actually dramatic enhancement within the exercise animals. And we also see some learning curve differences, but we actually see that it's not the same as what we see for the hippocampus sensitive task. In this case, we see that the exercise animals start out better on the response learning task and then the sedentary animals, but the sedentary animals end up eventually catching up to the exercise animals. And this is an important thought or finding to hold in your thought, in your mind before, until we get to the some later findings. Okay, so then the next question is whether cognitive priming can enhance different kinds of learning. So we know cognitive priming is part of learning itself, but can it enhance different forms of learning? So what we did, and this is sort of a complicated slide, but it's a very simple, straightforward study. We tested rats on learning tasks with or without prior working memory testing. So instead of exercise now, the animals had 20 minutes, just 20 minutes on a working memory task that we call spontaneous alternation. And in this test, rats have to use the cues in the room to visit different arms of the maze. Now, what's really interesting about this is they don't need food motivators to do this. They're naturally motivated to navigate this maze. And they show alternation behavior. And again, I'm not going to show those results, but they do show substantial alternation behavior in the animals that were primed. And our controls were actually unprimed, just held in their home cage in the room for the 20 minutes. They just weren't put on the maze. So they had the same experience except for the maze training. And then we looked immediately 1 hour and 24 hours later, so 3 time points, again, in different animals, and we tested them either on place or response tasks. Now, I'm just going to show you the results from the 1 hour. It's the most robust finding, and we use it for our future studies. And just let me say that we actually saw effects on both place and response tasks at all 3 time points. So here, again, very simple, straightforward findings that the rats that had the priming with the working memory task, the spontaneous alternation task, showed improvements on the place learning task, and also improvements on the response learning task, okay? So, and this was 1 hour after priming. So the effects lingered for at least 1 hour, and as I mentioned, we saw the effects for 24 hours. We didn't test longer time points yet. Okay, so I just told you that physical activity can enhance learning. So these animals are navigating a maze. So in order to rule out the fact that the exercise on the maze for 20 minutes was what was actually enhancing the memory, the learning on these tasks, we actually did a study where we primed the animal instead of with a 4-arm maze, with a 2-arm maze that was converted into just a straight alley. So the animals just shuttled back and forth between this arm and this arm. And then we tested them 1 hour later for their ability to learn place and response tasks. Now, I want to mention that during this 20 minutes of testing, they entered the same number of arms as they did on the 4-arm maze. So they were the same activity measures. And what we see here is, lo and behold, that when they're engaged in what we call a task that might have lower cognitive load, that's not requiring as much engagement by the animal, we actually see that there's no enhancement by priming on the 2-arm maze, okay? So we lose the priming effect if the cognitive load is dropped off. So it doesn't seem to be simply due to just movement on the maze. Okay, so of course, because we're interested in neural mechanisms, we wanted to next identify some kind of neural mechanism that may be involved in these priming effects and that may be shared with between physical and mental activity. So we know that actually the work of many others, that Bill Greenough and his academic offspring and many, many others, that physical and mental activity can increase brain volume, can increase neurogenesis, can stimulate the development of new capillaries, angiogenesis, and blood flow. In fact, this is one of the, blood flow. It can promote dendrogenesis and synaptogenesis, and it can modulate neurochemistry. Now, in the lab, we do a lot of neurochemical measures. And so again, there are many different measures that can be modulated by physical and mental activity. And we look at metabolism, but I'm not going to have time to talk to you about that. But we also are interested in trophic factors, particularly BDNF, brain-derived neurotrophic factor. And this one in particular, because actually what's interesting is BDNF is involved in a lot of these kind of other, what you might call upstream, not really upstream, but upstream from this on the slide, effects of physical and mental activity. So BDNF becomes a very good candidate for the shared effects. So as many of you probably know, BDNF is a small protein that belongs to a family of growth factors called neurotrophins. And its mature form is supposedly the most studied form, but it's people think it actually is kind of pro-neuroplasticity. And it's cleaved from a bigger peptide called the propeptide. Now, pro-BDNF also has its own actions, as you'll see in a little bit, but we've been studying mainly mature BDNF until recently, when we can actually measure in our neurochemical assays both pro and mature, which we started to do recently. Mature BDNF signals through tyrosine receptor kinase B receptors, or TrkB receptors, to induce cell signaling pathways that are involved in plasticity and, as you hopefully will see, learning and memory. It's released either pre or postsynaptically, depending on the structure of interest. It engages in constitutive and activated release, so activity-dependent release, that is action potential, sort of neural activity-dependent release. The levels are actually high in the hippocampus, the cortex, the striatum, basal forebrain, and other areas in the brain, but highest in these. And it's actually also found throughout the body. So it's found in the reproductive tract, it's found in adipose tissue, in muscles. It was just first identified in the brain, and so that's why it's called brain-derived neurotrophic factor. And then it's also increased following regular physical activity in the brain and periphery, and people have shown this in humans as well. And of course, as many of you know, it's known for its ability to stimulate neuroplasticity, so it becomes a really good candidate for us. And this is really just to show you that I'd like to show you that just to make 3 points about this. This is a very schematized version of BDNF, showing you it has a pre-region, a pro-domain, and a mature domain. And I want to make 3 points about this. First is that the BDNF gene gets cleaved, as I mentioned, into the pro-domain and mature domain, but and it gets packaged into vesicles as a pre-pro protein. It gets cleaved in the vesicles, some of it gets released as pro-BDNF and mature BDNF, and the pro-BDNF can actually do its actions through the P75 neurotrophic receptor or the sortilin receptor, or it can be cleaved in the synapse into mature BDNF. So there are many different actions, cleavage products. Now, the second point is that pro and mature BDNF signal through these different receptors, the TrkB versus these other two. So you can use these receptor activations or manipulations to the receptors to dissociate the effects of pro and mature BDNF. And also, we have evidence that BDNF signaling through its TrkB receptor is what mediates the BDNF contributions, as I'm going to tell you about in a minute. Now, we have not tested these other receptors, but we have tested the TrkB. So the first thing we wanted to know was that if BDNF is involved, the simplest approach is, does it increase with the priming activities that we're interested in? So we have shown, yes, that 3 weeks, like others have shown before, 3 weeks of exercise, voluntary exercise, increases BDNF in the hippocampus and in the striatum. Okay, and this isn't necessarily new. Others have shown this, especially with mRNA. So expression levels go up. Now, what's interesting is we don't have very tight control over the timing. So we don't know when the animals last exercised. So we don't know how recent their exercise was. And we also show that animals trained on just 20 minutes of the spontaneous alternation task show an increase in BDNF in the hippocampus and striatum as early as 1 hour after the task. So we see an increase in tissue levels of BDNF. Now, we actually see an immediate increase in the hippocampus, not in the striatum. So it might be surprising that the BDNF levels are increased so quickly in these structures. But remember that we're looking at mature BDNF, and it could be due to a cleaving from the pro-BDNF to the mature. So it might not be that synthesis is occurring immediately, but it may be the processing of BDNF that is occurring this rapidly. Okay, so what we found is that physical and mental activity improved place and response learning, and that physical and mental activity increased BDNF in the hippocampus and striatum. Okay, so pretty straightforward. So the next thing we were interested in is whether increased BDNF supports experience-dependent improvement through its signaling actions, acting more as a neuromodulator, or through its trophic actions, acting more as a neurotrophin. So now the alternatives are sort of laid out here. If it's acting as a trophic factor during the priming, let's say during exercise or during that 20 minutes of spontaneous alternation, that then the increased BDNF would be actually producing the substrates for plasticity. And those substrates for plasticity in turn are producing the priming effect, the benefits to place and response learning. So if this is true, you wouldn't need any increased BDNF during learning, okay? It's all done, its actions are all done during the priming. If BDNF is acting through its signaling, cell signaling actions as a neuromodulator during learning, then the idea is that the signaling itself is producing the priming effect, then you would need BDNF signaling during learning in order to see the benefits of exercise and mental priming. So we tested this by blocking the receptor for mature BDNF by blocking TrkB receptors with a compound just before learning. So the priming event is over. So they've had all the time to have the priming do its work. And we block it just before learning to see if blocking BDNF signaling impedes or reverses the priming effect. And so what we did is, so the first study was done with exercise, where we take animals that were assigned either exercise or sedentary, implanted them with cannulae, and then just before testing, 30 minutes before testing on place, we infused the hippocampus with the TrkB inhibitor. And in a separate group of animals, we infused 30 minutes before response training, we infused the striatum with the TrkB inhibitor. Now, the compound we used was K252A, and it was compared to the effects of the vehicle DMSO. And so I'm going to show you the results here for exercise. So for place learning, we see the expected, even in animals that had cannulation, we see the expected enhancements in learning with exercise. And then we actually see, if you look here, animals that had exercise but received the TrkB blocker during learning actually attenuated this enhancing effect. So this suggests that we needed to see to have BDNF signaling during learning in order to see the exercise effects on place learning. Now the same, so for response learning, we actually see this, this anticipated benefit of exercise in vehicle-treated animals. And then once again, we actually see the attenuation of that priming effect in animals that had the BDNF receptor inhibited. So you need BDNF signaling during the learning itself in order to see the enhancements, which suggests that it's the BDNF signaling that's important, not necessarily not only the trophic effects. So just here, just to show you this, we have this kind of surprising effect that the blocker alone, at least for response learning into the striatum, seem to enhance learning somewhat. Now, we don't see differences between the primed animals and the unprimed animals with the blocker. So the good news is that the K252A alone is not impairing learning, thereby producing additive effects of exercise and the blocker alone, but instead it interacts. So it is enhancing, and we really couldn't figure this out. One idea is that it has some off-target actions that may actually enhance memory, but that feels a little bit like speculation. So we haven't really tested this, but hold that thought, because I'm going to show you something at the end that has made us think again about these results. This was surprising. We didn't expect to see enhancement with the drug itself. So then we tested this to ask the question is whether the TrkB signaling during learning was necessary in animals that were primed with mental activity and not exercise. So we did the same design, but in this case, the animals were primed on the spontaneous alternation task, or were the untested controls, and then tested on place or response tasks an hour later, like I showed you before. And 30 minutes before testing, they received hippocampal infusions or striatal infusions before testing on the canonical task, before place or response learning. Now, in these studies, we did not test the non-canonical actions. And so what you can see here is the expected enhancement in place learning with spontaneous alternation that we've seen in the past, even with animals that had cannulae implanted with vehicles. And again, we see the attenuation of the priming effect of spontaneous alternation in the tasks that receive the TrkB inhibitor. These data are remarkably kind of uncannily similar to what we've seen with the exercise effects. And likewise, for response learning, we see the same, the enhancement with the spontaneous alternation, and again, the attenuation with the TrkB inhibitor. So collectively, what we've seen is that BDNF signaling during learning was necessary to see the benefits of both physical and mental activity on both hippocampus sensitive place and striatum sensitive response learning. So this is really the first bit of evidence that that BDNF was a shared mechanism, that BDNF signaling during learning was a shared mechanism. Okay, so the question is more BDNF released during learning in the hippocampus and striatum of primed rats? Now, we would want to show that because that would actually suggest that it actually is being released and doing its effects. Now, this is actually difficult because no one has ever looked at this before, because it's really, really difficult to measure large molecules using microdialysis, which is the technique that we use in the lab. And so, so we really don't know. All of the work suggesting that BDNF is released with activation has been done in vitro, either in slices or in cell cultures. And so Claire, as I mentioned, undertook the task of modifying our microdialysis methods to measure this large molecule. It's a small protein, but it's still a large molecule. So what she had to do is we had to use very large pore sizes for our microdialysis probe. But in doing so, what we had to do is we actually had to slow down the microdialysate to avoid abnormal activity in the brain, such as seizure activity and so forth. And then to slow down when we slowed down the perfusion rate and increased the pore size, it was very difficult to collect the sample through the narrow tubing. So what she ended up doing is adjusting the outlet to a special kind of pipette tip, literally, so that it would change the capillary action and draw the fluid through the microdialysis probe. So I only mention this because it really took her probably the good portion of a year to optimize this method. So don't get discouraged, for you students out there, if you're really motivated to try to ask a question and the only technique you have is to create something, put your nose to the grindstone, as they say, and keep on plugging on. So she actually was able to collect the sample, and she actually also optimized our ELISA method so she could actually measure samples in the time period that we would want for our experiments to look at in vivo release, both in the hippocampus and the striatum. So this just shows you her timescale for the studies I'm going to describe to you, where she starts the dialysis after she implants a probe, and the first 6 hours just junk sample as a probe equilibrates. And then she collected for 90 minutes while the animal, before the animal was put on the maze. And then she collected for another 90 minutes while the animal was being trained. So she had to actually also modify her training a little bit. And then she had a 90-minute post-training. Now, this was also that she could collect enough sample to run one assay per animal, per time point per animal. So you can imagine if it didn't work, she had to go back and start over again. So the experimental groups here, because the spontaneous alternation priming, the mental activity priming was a little bit harder to work out, we started with exercise. And here are the experimental groups where you see that there are 3 groups, sedentary. So we have animals that had hippocampal microdialysis probes and striatal microdialysis probes to collect extracellular fluid to measure BDNF in the extracellular fluid. And we had animals, since no one had ever done this before, we were really interested in identifying if there was a double dissociation in the release of BDNF while animals were on these different tasks. Again, no one had ever done this, so we didn't know what to expect from BDNF release while the animals were on the task. So the first set of slides I'm going to show you is just the memory systems part of this. And then I'm going to show you the priming with exercise, again, on the canonical task. So whether exercise can prime the BDNF release when animals are on place in hippocampus, or whether they can prime the BDNF release in striatum when they're tested on response learning tasks. So hopefully that's clear. So the first I'm going to show you is whether just the multiple memory systems part of this. So if you look on the left here, you can see that these are recordings of BDNF, so sampling from the hippocampus. And what I have is in the purple, the squares are animals that were trained on the place learning task, and the diamonds in teal are the animals that were trained on the response learning task. And this is relative to baseline. It's just the easiest way to express these data. And so what you can see is that animals trained on place, you see a slight elevation. It's not significantly different, but a slight elevation in extracellular BDNF when they're trained on place learning. That comes down after training. It's quite variable, however. Interestingly, what you see is that in the hippocampus, when animals are trained on the other tasks, the non-canonical task, we see a dramatic decrease in BDNF when in the hippocampus, when they're trained on the response tasks. And that this interaction is significant. So we see a bidirectional effect depending on whether they're trained on the canonical task or the non-canonical task, suggesting that there may be some competition going on here. In the striatum, we see the similar effect, that when they're trained on the response learning task, which is the striatum sensitive task, we see an increase in BDNF being released. However, when they're trained on the place learning task, the non-canonical task, we see a decrease. So now there's this beautiful double dissociation between BDNF release in the hippocampus and striatum during maze training. So the next question is, what does exercise do to this release? What we've seen, I'll show you first, and the top is what happens in the hippocampus. So these are the results I just showed you above, but shown as the actual levels that are released during training. So these are picograms per mil of BDNF. You see that the sedentary animals, which I just showed you, you see the exercise animals start out at baseline the same as the sedentary animals, but that they have training-induced potentiation of BDNF while the animals are trained that comes back down to baseline. So we see a potentiated response while the animals are being trained. In the striatum, we also see augmented BDNF release due to exercise status, but a very different pattern emerges. In the striatum, we see that the animals enter the task at baseline with higher levels of BDNF. But we don't see a large, in the exercise animals, we don't see a large training-induced increase in activity-related BDNF release. Okay, so it kind of stays the same throughout training. And in the sedentary animals, as I showed you before, it increases, and actually, when it's graphed this way, we see that there's actually continued increase in the sedentary animals. So a very different pattern of effects that might actually reflect, and this is a speculation, activity-dependent release potentiation in the hippocampus and potentiation of constitutive release in the striatum. Now that's very tempting and also very speculative. We haven't tested this yet. But what I want to do is then remind you again of the results from the learning task that I showed you before. And again, I'm also very tempted to say that the BDNF during learning is very similar to the accuracy of the behavior during learning, okay? And actually, sorry about that, my mouse just fell off the pad. And actually, if you just kind of plot the data the same way, you can see that you can start to see that the patterns emerge, that where the BDNF release looks a lot like the learning curves, okay? So it's tempting to conclude, or at least as I kind of said, speculate that the BDNF changes actually drive the behavioral changes. But of course, it's entirely possible that the BDNF changes simply correlate to the changes in behavior. And we haven't continued this recently, but we'd like to further this work to ask that question in particular. Okay, so far we found that in the hippocampus, exercise facilitated task-dependent elevations in BDNF, in the striatum, exercise enhanced BDNF levels before training. So what we'd like to do is we still need to test if the same patterns emerge with the spontaneous alternation priming. And as I mentioned, we have to finagle our sampling methods a little bit in order to allow us to get we haven't done this yet. Just let me leave it at that. Okay, so the question then remains is, so if we see this, so if we take the results with the striatal infusions, the question that we next ask is whether pre-training infusions of an agonist to the BDNF receptor, to TrkB, would enhance response learning. Because if, in fact, there's more BDNF at baseline in the striatum that's contributing to its faster learning, then if we infuse BDNF agonists just before learning, we should mimic the effects of exercise. And so what we did, and this was the work done by an undergraduate honors student in the lab, we're preparing this for publication, hopefully in the next several months. But what he did was he actually took a BDNF mimic, it's called 7,8-DHF or dihydroxyflavone, and he infused it bilaterally into the striatum 20 minutes before the animals were tested on a response learning, the striatum sensitive response learning task. So simple design, but to mimic the effects of exercise. And what he found is that, lo and behold, yes, all the doses that he chose actually did enhance learning, as seen by a decrease in trials to reach criterion. In fact, we didn't see a very dose-dependent changes, and we might need to go lower and higher to see the normal U-shaped function that we typically see. Now, what I'd like to show you, though, is that his actual learning curves, looking at percent correct, actually look a lot like the exercise effects on the response learning task. So yes, the animals with the most effective dose, the 1 microgram dose here, actually showed better learning in the first trial block, and the vehicle animals actually caught up by the end of training, very similar to the exercise effects on striatum sensitive response learning, as I just said. So we think that this supports the idea that there's an increase in basal amounts of BDNF in the striatum in animals that are primed. And again, we're hoping to keep continuing to test this question. Now, BDNF can signal through its TrkB receptor through many different signaling pathways. And I know this is complicated, not as complicated as many cell signaling pathways are. But I just wanted to show it that there are many pathways that are involved in metabolism, synaptic plasticity, neurotransmitter release, cell survival, and neural protection. Okay, and so I want to focus your attention on one of these pathways, and as I mentioned before, this GSK3 beta kinase that we've become interested in. Now, this kinase is known for its ability to stimulate apoptosis, its ability to hyperphosphorylate the tau protein, which we know is involved in many neurodegenerative diseases. There are tauopathies where the hyperphosphorylated tau is the problem. We also know that it blocks transcription factor activation, including CREB activation and many others. Well, interestingly, signaling through the TrkB receptor through the PI3 kinase pathway and downstream AKT, through phosphorylation of this enzyme actually inactivates the enzyme, thereby promoting survival and plasticity, perhaps by decreasing these sort of negative actions of the GSK3 beta. So phosphorylation actually inactivates this enzyme. So priming may inhibit glycogen synthase activity through BDNF signaling, thereby producing its effects. So we've started testing this idea by actually looking at GSK3 kinase phosphostate or inactivation state in different conditions. So in conditions that activate the TrkB receptors, such as what I just showed you through bilateral DHF infusions. Now these are in untrained rats now. So they were just rats that had infusions, and then we sampled the brains and ran the Western blots to show GSK total amount, the phospho amount, and then the ratio. We see that in animals that received DHF infusions, the 1 microgram dose, we see an increase in phosphorylation state. in these animals and in the ratios, suggesting that there's an increased inactivation in GSK3 beta when we use this BDNF mimic, okay? Now, what about in situations where we've shown that we see increased BDNF release, such as in response training? So if we take animals that were unimplanted and were trained on this response task, and we then looked at the phosphostate of GSK, we see that, again, that simply engaging in response training inactivates this enzyme, this kinase. So we think that that might be a way that cognitive activity may actually lead downstream to later benefits to subsequent cognitive activity. So this might be one of the mechanisms downstream from BDNF release, downstream from TrkB activation for our priming effects. Now I want to end with just one tidbit, and I know it's coming close to the 1 hour time point, but we really literally just got these data off, like hot off the press, and it's very preliminary, and I usually don't show data with only ends of 2. So I'm this full disclosure here, but I have to show this as we've been working for the last couple of years on a model of a single nucleotide polymorphism that I'll explain in BDNF that I'll explain. So the question though is that in conditions that prevent BDNF release, should prevent the priming effects if this whole cascade of events really is one of the mechanisms for the priming effects. So I've already shown you that priming seems to benefit place and response learning through this BDNF signaling. And the idea is that there is a single nucleotide polymorphism that's found in humans. It's called the Val66 MET. And it actually produces a Val-MET switch in the pro-domain of BDNF. And this actually changes the way BDNF is packaged into vesicles, and it actually limits its activity-dependent release. And this has been shown, again, only in vitro and in sort of indirect measures of changes in BDNF in serum and so forth. Now, the Val version, the Val allele is actually the more common allele for this polymorphism, at least in the United States. It varies in different populations. The MET allele is the less common, okay? So it's been found that MET carriers are actually at higher risk for various neural disorders. They're at higher risk for anxiety disorders. And I know that you are interested in, some of you are interested in emotionality and the whole idea of how that affects learning and memory. We know that they have increased anxiety disorders, increased risk for severity of Alzheimer's disease, decreased sensitivity to treatments in Parkinson's disease. Women that carry this polymorphism, the MET allele, are actually at greater risk for getting Alzheimer's disease and may actually be insensitive to estrogens, as we've been testing in the lab. So what's really great, though, is that my colleagues at Michigan State University have actually developed a CRISPR-Cas9 model of this in Sprague-Dawley rats of this single nucleotide polymorphism, where they've simply replaced one nucleotide to create a Val-MET switch at the same position, relative to position in the rat's BDNF gene. And it's a Val68 MET. It's 2 amino acids different, but it's the same position and it changes packaging. So we actually have been using the females they're females, they don't use females. And so we have a whole study that I can talk to you the next time I come to Mexico about that work. But they were actually closing down their breeding colony, and they had a cohort of a few males, and they asked me if I wanted their males. So they sent them to us. And I said, what better way to test our priming effect in the male Sprague-Dawley rats that we've been using, where we can actually look to see if rats that have deficiencies in BDNF release, are they insensitive to the priming effects that we've seen? So I have one tidbit. I told you it's just a tidbit. We actually found that priming, so this is with mental activity now, priming with the spontaneous alternation working memory task is not seen in rats that carry this polymorphism. So here you see, if you look on the right, these are what would be considered the wild type animals, or the more common variant. And you see that just like we did before, and this is the work of a postdoc, Bob Gardner, that animals that were primed actually showed better learning on the place learning task. We didn't have enough animals to test the response learning task. Interestingly, priming failed to improve learning in the Met rats. Now, one of the reasons it might not have improved it is that they're already doing so well. So learning was faster in the Met rats than in the Val rats, which was really surprising. We were shocked to see this because these animals supposedly don't have BDNF release, activated release, or have blunted BDNF release. So this tells us that blunted BDNF release did not impair their learning of the task, but it interfered with the ability to be primed by a prior experience. And of course, this is just hand waving, but this suggests that there's something else going on that's facilitating their basal learning rate. And if you remember in our studies where we blocked BDNF signaling by blocking the receptor, we also saw good learning in those animals. So even though we actually saw a impairment in the priming effect, as we see here, when there's impeded release or signaling, we don't seem to see generically the problem in learning. We just seem to lose the priming effect. And we're really curious about this. And this has really been bothering us. And so we hope to have an answer to this in future studies. So our current directions are to model menopause and neuroprotection by estrogens. As I mentioned, we'd like to extend this to, and we're doing this now, extend some of our questions to females. So we're modeling this in the CRISPR-Cas9. We're looking at sex and hormone status effects on BDNF and its role in cognitive aging, and we have some data on priming in old animals, but I wasn't able to show that here. And we'd also are starting to look at linking the BDNF signaling to metabolism and cognitive aging, which is another line of work in the lab. And so these are the same take-home messages. I added blunted BDNF release prevents priming because that was just the tidbit I just showed you. And so hopefully I've showed you that, convinced you that there's a shared neurobiological mechanism between physical and mental activity, and that mechanism is BDNF signaling. And so I want to leave you with one take-home message, and that is to really just be a user and boost it. And that's my request to you. And then before I sign off, I'd like to acknowledge the current members of my lab, the past members, especially those that did the work with exercise. The lab, this was winter 2020. This was just pre-COVID, showing, collaborators, Paul Gold, Kathy and Carol, who are my collaborators at Michigan State. The funding, of course, we've been, as Gina mentioned, I'm in my last no-cost extension year for my NIH grant. So I've been applying, kind of chaotically over the last year. And then I'd like to also thank the SU animal care staff, because we couldn't do our work without them, and then the animals themselves that have kind of dedicated their lives. And then just to show that we do have fun when we can all get together, but that this is kind of the new normal of things. And so with that, I'll leave you with, this is our current lab. So let's leave you with that. So, and I'll take any questions that anyone might have, and I really appreciate your attention.
Thank you so much, Donna. It has been a wonderful talk, and we have some questions. A very general question, I must say, is a comment. Donatio Mungia, he said that given the benefits of physical activity, is it unethical not include exercise protocols on the housing guidelines for laboratory animals? What do you think about it?
Yeah, no, I actually think that's really interesting. And there's Chris Comer, who I think he has since retired, but he was at University of Illinois at Chicago. And he, so I'll tell you what I think. I think perhaps, yes, after everything we've seen, the animals do need to run. They do need to move or have some kind of enriching experience. And so the problem is that how do we start that and not, so if anything, you could argue that we always need to have an enriched condition in our experiments, but maybe because we have so much, we have decades and decades of results, not that that justifies not doing it, but decades of results that actually tell us about animals in isolation and what their brain is like. And we don't have as many results in enriched environments. So I think we have to at least catch up so we know what those enriched environments are doing. And perhaps we need to flip it so that we can look at the isolated environments as the experimental condition. Hopefully one day we can get to that point and the enriched environment is the standard condition. Now, if you could argue we could put a running wheel or some kind of environmental enrichment, and some animal care facilities, some institutions require that. And I think NIH has actually, at least in the United States, NIH has considered requiring some kind of enriching. They have required, we need to have an enriching, like a chew toy in the cage, but that's all. And it's not changed every day. And we know that enrichment really works if you change the toys. So just having the same toy every day might not really be enrichment the way we think about it. So I'm not sure I answered your question, but I think for me, it does pain me to see the animals in isolation. In fact, I think my biggest ethical right now in this stage of my life, the biggest ethical concern I have is not the humane euthanasia that we use in killing the animals, but actually housing them in such isolation.
Related to this question, there's another question where Jesus asking, why did you use the voluntary exercise model instead of the force model?
Yes, that's a very good question. And so we were trying to, first of all, the force model is great because you have control over the exercise, but there are problems in itself in that it can actually cause a heightened stress response. And so some of our work looks at the interactions of reproductive hormones and maybe adrenal hormones. And so we didn't want to introduce the heightened stress response, even though we lost some of the control over the running behavior. That said, one of the problems with doing priming in our old animals is that they don't run voluntarily as well as the younger animals. So in those cases, we've actually have the plans to use a forced exercise paradigm where we start them learning to use the treadmills by just walking, and then up to more of a running manipulation. Now, the other idea is that with humans, even in studies, humans that engage in exercise tend to engage voluntarily. So the voluntary exercise does provide a good translational model for what we see in humans.
Oh, I see. Thank you. So there's a lot of questions. So the next one is from Mauricio Diaz, our secretary of the Institute. And he said, thank you very much for your interesting talk. It is known that using the running wheel has an important impact in circadian physiology. Do you think that this possibility could play a role in your experimental model?
Yes, I actually think that was a very interesting idea. And we know BDNF actually has been shown to be involved in sleep drive and in sleep quality. So if at that level, yes. And it could be that the running is actually increasing sleep quality, which then in turn is producing the benefits. However, the fact that we see, if we block the BDNF we block the benefits of our priming of exercise and mental activity, the priming with BDNF blockade during learnings. So we don't think it's the only story. So it could be that the better sleep is actually increasing the BDNF, which then there's increased signaling during the learning to promote the better learning. So sleep quality could be in between the exercise effects. It's unlikely that it's between our mental activity because there's only an hour between the two, and the animals do rest in their cages, but we haven't actually measured sleep patterns in those animals, and that would be an interesting study to do, or disrupt the sleep to see if you then lose the effects of the priming.
Of the exercise, yeah. Thank you. So there's Roberto that wants to have the voice. Rosario, ¿le puede dar la voz a Roberto?
¿Me oye? ¿Me
Sí,
oye?
te escuchamos.
Okay. Sí. Hi, Dana. Thank you for this wonderful talk. Very nicely done. I have a couple of questions. Is there a differential role of medial versus lateral dorsal striatum in the effects that you presented?
Yeah, we have not evaluated that. So we've actually been looking, I didn't mention this, and I should have, because we have the aficionados in the audience too, but that we've been targeting pretty much dorsolateral striatum, and definitely not sort of ventral striatum or medial striatum. And I do think, given some of the work, I mean, certainly the work of like Mike Ragazzino in terms of different kinds of task switching and so forth, but also in terms of, I mean, others looking at the role of ventral striatum, we have, it would be interesting, but we haven't tested that at all.
Okay, thank you. Well, by the same token, how about dorsal ventral versus ventral hippocampus?
Oh, well, that also, so we've, so interestingly, when we have done our infusion studies, we infuse into the dorsal hippocampus. However, when we did our BDNF studies, simply because of the size of the probe, just for the logistics of the experiments, we had to move more posteriorly, and we had to actually collect samples from the ventral hippocampus. So we actually have some slightly different, different techniques or different sampling sites for our different results. However, it seems that it's very consistent. So we've only tested in sort of the blockade in dorsal hippocampus. When we do our measurements, we take out the whole hippocampus, and we haven't dissociated dorsal versus ventral. Now, it's interesting because it would, the competitive interactions between striatum and dorsal versus ventral and hippocampus may be very different. So we might expect to have different results if we did look at that, but we have not yet done that. So if you have any suggestions, I would appreciate it.
Well, maybe a little later. Thank you very much, Dana.
Thanks, Roberto.
Well, there's another question from Dr. Fernando Peña. And he said, which cells are releasing BDNF in an activity-dependent manner?
Oh, that's a really good question. So, so we think that the pyramidal cells, so some really beautiful work of Chris Gall and others, I'm just blanking on the names right now, has shown in vitro slices and in cell cultures, like if you actually look at primary cell cultures of hippocampal pyramidal neurons, you can actually use potassium and show that potassium increases the BDNF in the medium. And they've also done some very nice kind of molecule following to show that it seems like it is being released from the pyramidal cells. Now, in the striatum, the cortical input to the striatum is that those axons are most likely what release BDNF into the striatum. Now, in the hippocampus, what's interesting is that the dendrites can release, the dendrites can release BDNF, as can the axons in the hippocampus. And so there's probably entorhinal cortical release as well in the hippocampus.
Thank you. There's another question from Cristina Medina. And she said, thank you for the interesting data in the presentation. I saw that there is less raw ECF and BDNF in the striatum compared to the hippocampus. Do you know why?
We do not. And so that's a really good question, a very astute observation. So I didn't mention that, but the striatal levels are about a fourth of the levels in the hippocampus. And one possibility is just the cell density is a little different in the striatum. And so it might be the packing cell density and not that the amount of BDNF being released from each cell is different. Again, if it's from the cortical input, then it wouldn't, it could just be that the cortical input, is different. It also could be that, we don't know. We have no idea why that would be so.
Okay. Well, thank you, Donna. I think that's all for today. And I hope in the next future you can come and not also give a talk. Also after the talk, have lunch with us in a place here in Querétaro. So I really thank you for your talk and thank you for the audience to be here. So.
Well, I'd like to thank everyone for your, continued attention. I know being on Zoom for an hour plus is not always easy, so I appreciate it. And thank you for your interesting questions. I have a lot to bring back to the lab to tell them to get started on. So thank you.
Yeah, and I'm sure that if someone wants to write you and have interest in your research, they can do it, I think. So we can find you very easy.
Oh, absolutely. I had my email on the first slide, and anyone, if you Google, I think you can Google me and find me. So please, I love staying in touch with people. So.
Well, thank you very much. Thank you very much, everybody.
Everybody stay well, and hopefully I'll see you at a conference someday.
Hopefully.